Hello everyone, my name is Khushal Ingde and today we will learn about the bacteriophages. In this today's topic, I will describe the structure of T4 phage, its types, morphogenesis and kinetics. So first of all, we will discuss about the structure of T4 phage. So friends, as we know that T4 bacteriophage is a large virus and stores its genetic information in the form of double-stranded DNA molecule. This DNA is packaged inside a proteinaceous head. The size of this head is about 78 into 111 nanometer and it contains contractile tail which measures about 113 into 20 nanometer. Its genetic material is around 168,800 base pairs long and contains about 150 genes. Apart from the head, the other important part of this virus is nothing but the tail. The tail of this virus contains several important compartments. Its central hollow core provides the channel through which the phage DNA is injected into the host bacterium. The sheath of this tail functions as a small muscle. This sheath contracts and pushes the tail core through the bacterial cell wall. Then, the phage also contains the 6 tail fibers. These fibers are used to locate receptors on the host cell or the bacteria, and the tail pins on the base plate then attach firmly to these receptors. Hence, if any phage wants to infect an E. coli cell, then all of these components must be function correctly for the phage to infect an E. coli cell successfully. Now coming to the next important topic, that is types of bacteriophages. So as we know that the bacteriophage which is infecting the bacteria is maybe of two types, that is first one lytic or virulent phage and the other lysogenic or the temperate phage. So coming to the first type, that is lytic or virulent phage. These phages are phages which can multiply only in bacteria and kill the host cell or lysis at the end of this life cycle. Lysogenic or temperate phages are those that can either multiply via the lytic cycle or enter a quiescent stage in the cell. These bacteriophages multiply through lytic cycle and follows the sequence, that is first attachment, then penetration, biosynthesis, maturation and release. So during the first phase, that is attachment, bacteriophage attaches to the host cell or the bacterial cell. After successful attachment, it will penetrate bacterial cell and inject its DNA within that cell. Then during third step, the injected phage DNA will start the biosynthesis of various proteins which will make viral components of that phage. During fourth step, the viral components assembled and new phages are synthesized, and in the last step, host cell breakdown or the lysis takes place and new phages are released into the environment. Now, these phages are again ready to infect the new bacterial cell or the host cell. So, all these events takes place during the lytic cycle. Now, about the morphogenesis. As we know, T4 bacteriophage is a lytic phage. When it infects bacterium, it replicates and kills the host cell and produces near about 300 progeny viruses per infected bacteria or the cell. After the phage DNA is injected into the host bacterium, it quickly, means within 2 minutes, directs the synthesis of proteins that will shut down the transcription, translation and replication of the bacterial genes. This condition will allow the virus to take control of the metabolic machinery of the host cell. Some of the phage genes encode nucleases that degrade the host DNA. The other phage contains or the other phage proteins initiate the replication of phage DNA. Later, the genes that encode the structural components of the virus are expressed. Thereafter, the assembly of progeny phage begins. Infectious progeny phage start to accumulate in the host cell at about 17 minutes after the infection. At about 25 minutes after infection, a phage-encoded enzyme called as the lysozyme will degrade the bacterial cell wall and ruptures or will break down the host bacterium, which will release about 300 progeny phage per infected cell. T4 phage encodes nucleases that will destroy or degrade the host DNA. The degradation products are then used in synthesis of the phage DNA. But these enzymes degrade host DNA without destroying the self DNA or the phage DNA. The bacteriophage T4 DNA contains an unusual base, that is 5-hydroxymethyl cytosine. In short, it is called as the HMC. Here, cytosine with a CH2OH group attached to one of the atoms in the cytosine molecule instead of cytosine. Derivatives of glucose molecules are attached to this unusual base, that is HMC. These modifications protect T4 or bacteriophage DNA from the degradation by the nucleases that is uses to degrade the DNA of the host cell. In this way, it will protect its genetic material. 3 separate biochemical pathways generate heads, tail and the tail fiber, which are then joined to make a virus. A cluster of proteins forms the starting structure, that is nucleus, and the other subunits spontaneously arrange themselves around the nucleus in an orderly manner. Now coming to the next one, that is kinetics of the phage infection. So we know that first of all that bacteriophage will attach to the susceptible E. coli cell, and after 1 minute it will inject its DNA into that cell. After 1 to 7 minutes it will transcribe and translate the early genes, as well as it will block the bacterial DNA synthesis and will destroy or degrade the host chromosomal DNA. It will also block the transcription of host mRNAs and it will block the translation of host proteins also. Apart from this, small amount of the early proteins are produced from the early genes which are catalytic in function. Then transcription from the single phage genome will occur. Apart from this, during or after 7 to 15 minutes, replication of phage DNA will occur. After 10 to 20 minutes, translation of phage late proteins will occur. These will be the structural proteins. So this bacteriophage will need large amount of these proteins to build the new virons or the viruses. After 18 to 25 minutes, assembly of new phage particles will start, and after 25 minutes, lysis or the breakdown of host cell and release of the progeny will occur. It is called as the end of latent period. So it was all about the structure of T4 phage, its types and morphogenesis. Please share this video if you like and subscribe my channel for latest video, and also put your valuable comments. Thank you so much.